Okay, well, good afternoon, and thank you all for joining us on this snowy blizzard Science Cafe Thursday. I am Laura Parent. I'm Director of Strategic Partnerships and Research Development at the CTSC of Northern Ohio. Mackenzie Ritter is off today on a tropical adventure, and so we are all jealous of her for that. But I'm happy to be here with our presenter today. Our topic this morning is environmental health. And so, for those of you maybe not familiar with the CTSC, just a brief overview. Our goal is really to improve the health and of people who live in Northern Ohio by providing resources and services free to our university and hospital partners. Um, the CTSC is doing this through the help of our translational researchers and scientists and community members by focusing on kind of our key five areas and initiatives being resources and services, funding opportunities, community engagement, workforce development, and health informatics. Our presenter this morning is Dr. Bridget Hagerty, an assistant professor in the Department of Civil and Environmental Engineering at Case Western Reserve University. Her and her team use big data and multi-omic approaches to understand and engineer microbiomes of built and natural environments in order to preserve both human and environmental health. Dr. Hagerty's two major foci are shift in infrastructure to design and promote healthy microbial communities and improve ecosystem services through microbial community design. It's a mouthful. So thank you so much, and please join me in welcoming Dr. Hagerty to the stage.
Thank you so much for that introduction, Laura, and thank you all for being here. I've already got a brief introduction from Laura, but I wanted to give you a little bit about my background and how I come to be doing the work that I am. So, as we said, I'm Dr. Bridget Hagerty, and I'm an assistant professor in civil and environmental engineering. I also have a courtesy appointment in chemical and biomolecular engineering here at Case, and my background comes from a lot of different disciplines. And I had a postdoc in ecology and evolutionary biology, as well as in environmental engineering, and having both of those perspectives really informs the work that I do that cares about human health and the environment, as well as thinking about microbial communities in depth. And as Laura said, my research is really focusing on trying to engineer beneficial microbiomes, and this uses a lot of both lab and bioinformatic techniques to be able to hopefully have healthier microbial communities. We'll be talking a little bit more about that today. My PhD was in environmental engineering in a chemical engineering department, and my bachelor's was in biological engineering. And all of these backgrounds really inform what I do. So today we'll be focusing on how the environment impacts human health, and particularly the ways in which microbiomes are influenced by the environment and influence human health. And to start with, I'll do a brief introduction to microbiomes. If you're familiar with this research, it'll be a bit of a review, but I wanted to make sure that we're all on the same page. And then I'll talk a little bit about how microbes will be changing with climate change, talking a little bit about indoor air, and ending with some of my work on biodiversity. So to jump into things, just a couple of definitions. Microbiome is all of the microbes and environmental conditions in a particular environment. And if we just want to focus on the microbes, then we can talk about the microbiota, or all of the bacteria, the archaea, the viruses, et cetera, that are in a particular environment. And so if anyone wants to unmute themselves, I'm curious, what microbiomes are you all familiar with, or feel free to add it into the chat. Yeah, so I see gut, vaginal, skin, human, yeah, people talking about bacteria and bacteriophages. Cool. Yeah. So these are the ones that particularly in a health context we think about. But in my research, I'm also thinking about all of the other microbiomes that surround us. So thinking about those in the built environment, in the food that we eat and the water that we drink, in our food systems. And so all of these are interconnected and really critical to human health. And if we're trying to study microbiomes, increasingly we rely on these omics techniques. And thinking about back to basic biology or the central dogma, all life has DNA, which is transcribed to RNA, and then proteins are what actually do things in the cells. And we have different methods to study each of these. For instance, we can use genomics to study the genes of a particular organism, or transcriptomics to get in a window into what they're doing, or proteomics to see further what they're doing metabolically. And all of these give you a different window of both what cells are doing and who is there. And in my research, because I'm thinking about microbial communities, we do terms such as metagenomics and metatranscriptomics, which allows us, rather than focusing on a single organism, we can think about all of the microbes that are in a particular environment. And today I'll be showing some of my work that revolves around metatranscriptomics in particular, where we're looking at what are the genes that are being expressed. And so thinking about the connections between climate change and health, one emerging example is that of the rise of Candida auris, and there's thought that this might be related to climate change. And the basic idea is that because global warming is increasing global temperatures, more thermotolerant versions of C. aureus are evolving and are present in our environments, that those are coming into contact with us. And because with increased urbanization and contact between natural environments and humans, we're seeing more cases of C. aureus, and also that these fungi are actually more harmful. And so this is just one snapshot of how climate change is leading to changes in environmental microbes, and then that can lead to problems for human health. And there's lots of other ways that climate change can affect microbes. I'm not going to go through all of these examples, but I put this up to show that there's many different ways that that climate change might increase pollen production, which can lead to harmful negative health impacts, as well as lead to fungi to actually increase their spore production. And so in addition, climate change is also likely to increase microbial dispersal. All of these are obviously potentially problematic for anyone who has allergies. Having more mold growth is very problematic. As someone who studies the built environment, the presence and increased levels of mold is a particular concern. And so all of this that is happening outdoors can also have an impact on indoor air and air quality. So if we think about just a simple room, then if there's any moisture, then you can have mold or other microorganisms growing. Those can release toxins or their own spores into the environment. We as people are also continually shedding microorganisms. Our pets, particularly dogs, are also doing this. And then there's also exchange between the outdoors and the indoors. And in the indoor environment, as we're walking around, as we're resuspending microorganisms that might be in our carpet or on our hardwood floors as we're walking. And so all of these processes can be influenced by climate change. For instance, growth on wet surfaces is going to become more prevalent with higher indoor humidities as because of climate change, increasing temperatures, increasing humidity. Also, because climate change is increasing the likelihood of bigger rainfall events, then there's increased likelihood of water infiltration, which if you have more water, then there's more likelihood of fungal growth. And then, as with the previous table, if there's more pollen outdoors, if there's more fungal spores, then there's going to be new inputs from outdoors and also more input from outdoors. And just to stress the new inputs, as climate change leads to different microorganisms and different plants to be able to survive in different environments, then the specific microbes that we're being exposed to can change because our winters are becoming more mild. Today might be an exception, but in general, and so what species we're being exposed to is changing. And so all of this is to motivate the importance of understanding the connections between what's happening indoors and what's happening outdoors on human health. And another important consideration, besides just more fungal growth, is whether changes in humidity might be changing what fungi are doing. If we take an example from some pure culture work where some researchers grew Aspergillus flavus under different humidities, or here we'll call it water activities, they found that aflatoxin, which is a very dangerous compound, their gene expression increases and also the production of aflatoxin at higher water activities increased compared to at lower water activities. And so in this pure culture study, we can see that toxicity of this fungi is increasing, which led my PhD advisor to the question of, is it possible that in the built environment, that as you have more moisture, that you actually have more toxic fungi? And so that this might be explaining some of the reasons that we see that in some cases fungi is more harmful, and in other cases there'll be lots of fungi present in the environment, but it's actually not that toxic that people aren't getting sick. And so using transcriptomics to look at gene expression across an entire dust microbial community, we saw that there were systematic changes in gene expression with water activity. In this animation, you can see that all of the metabolism of these organisms, and I've called out some of the particularly important types of metabolism, so energy metabolism, carbohydrate metabolism, central things that all of life has. And as we go from these lower water activity, so like 50% water activity to 85% to 100% humidity, you can see that more of this map lights up. And this shows that these microorganisms are actually changing what genes that they're expressing when there's more water. And this is interesting scientifically, but from a health standpoint, this is really important because many of those genes that are being upregulated and are more expressed at higher water activities actually encode for allergens. And so what we saw in this community is that there's more allergen encoding genes being expressed, potentially because there's more general growth that's happening. And so there's more competition. And so the microorganisms might be producing more of these allergens to better survive in these good conditions. And so this is just looking at the production of the genes. It might not actually translate to allergenicity. And so we also looked at pure culture, where we saw that for Aspergillus fumigatus, when we grew it at higher water activities, the per spore toxicity actually increased. So this isn't just that there's more of the fungi growing, each individual spore is more toxic. And so if we're thinking back to climate change, that as we have more increasing humidity, higher temperatures, more water damage, then we might be seeing increased health effects. And so switching gears a little bit to think about diversity and urbanization, there's also emerging evidence that is linking together microbial exposures with immune development and other aspects of human health. And some of this research is very mixed, where some epidemiological studies are finding that as you have more green space, as in rural environments, you have less incidence of these immune diseases, especially autoimmune diseases like asthma and allergies, but other studies have found contradictory evidence where there's actually more incidence. So because of these conflicting results, there's a desire to have a better way of monitoring microbial exposures that we would have during outdoor activities. And so for this study, we took samples of the particulate matter on people's shoes during different walks in different environments. And so we focused along an urbanization gradient, moving from an urban gray environment that was just on pavement to an urban green environment where it's still in an urban environment, but it included some small urban parks and maybe some walk, not just on pavement, but it could also be on some dirt paths. And then finally, a rural green environment. And we were interested, does the microbial exposures change across these three environments? From previous work, we would expect this to be the case, but we wanted to see whether the shoe sole samples could actually replicate those results. And if one of my grad students, Sadia, showed that as you move from the urban gray environment to the urban green environment, that we actually have more gram-negative bacteria in the rural environment, which we would expect. And that the communities in these environments are very different from each other. So if we look at this NMDS plot, if 2 points are close together, that means that those communities are very similar to each other. And so if you just look close at it at a first pass, you can see that there's 2 clusters for each of the rural environments and that they're very different from each other. And that we see clustering for both of our urban environments with some separation between the urban gray and the urban green. And what this shows is that there's differences in the bacterial communities between the urban and the rural environments. We also repeated this for the fungi and saw very similar results. And so the next steps of this work and places where I'm looking for collaborations are to identify associations between these changes in microbial exposures and health outcomes. And so we're interested in both population level changes between rural and urban, but also individual. If you're living in an urban environment, but you go on lots of hikes, is that able to counteract what your day-to-day life is? And then also thinking still about the connections between indoor conditions and microbes and how does that actually affect occupant health. And so by understanding both these positive and harmful conditions, then hopefully we can better design the urban in our built environment to have more healthy microbial exposures. And so if we're thinking about translational science, then understanding the connections between microbes and the environment is an essential piece of environmental health. And if we understand these processes, then we can craft better policies that account for microbial influences on health and not only thinking about the negative impacts, but also the positive impacts. And as I said, as an environmental engineer sitting in a civil environmental department, this is hopefully will inform building design. And so thinking a little bit about funding, there's interest in the NSF to, especially within environmental engineering, to better understand how microbial communities change with the environment, both outdoors and indoors. And the NIH is also funding this work. They currently have a call for research connecting microbes and the built environment. NIEHS has historically funded this work. And in the past, some other agencies have funded more of the climate change work. Right now, that's kind of a dirty word. So being creative and how you talk about that is very important. And so that's where I just wanted to end it, and happy to turn it over to more discussion.
Great. Well, thank you so much for starting the conversation off with some really good research. I also want to take a second to introduce, we have a moderator with us today, Dr. Sarah Ronas. She is one of our CTS, Clinical and Translational Science Research Program awardees for her project, School-Based Research-Informed Asthma Toolkit for Healthy Elementary Students, or BREATHE is the acronym. The goal of the study being to identify and test evidence-based strategies to reduce chronic school absenteeism among school-age children with asthma. Dr. Ronas is also the director at the Center for Child Health and Policy at UH Rainbow Babies and Children Hospital. So thank you, Dr. Ronas, for joining us, and we will kick off kind of the Q&A and discussion portion.
Thank you so much, Laura. So the intention is for this to be very interactive. So those of you who are attending, please feel free to use the Zoom tools to raise your hand or throw a question in the chat. We will get this conversation going. I'm going to take moderator's prerogative. And because of my interest in asthma and the indoor spaces that children spend time, I was wondering if Dr. Heckerty, if I could put you on the spot to talk a little bit about your HUD work and the linkages between the microbiome and um, family exposures and experiences.
Yeah, I'd be happy to. So Sarah knows about another of my projects that I didn't talk about so far, which is I presented the work showing how there's these profound changes in the microbial communities in related to allergens and others, and all of that work was in chambers. And so we're really interested in understanding in real homes, can we see similar changes in microbial gene expression in the homes of kids with asthma? And the reason that we're really interested in this is that we know that the built environment affects asthma outcomes. Having high humidity can lead to worse asthma outcomes. Having mold obviously is problematic. Dust mites are problematic. And so we're interested in testing whether low-cost interventions to rectify those problems that increase humidity, that lead to mold growth, can improve asthma outcomes. And by understanding that, hopefully we can give caregivers more options for helping to address their children's asthma. There's not much that we can do about what's coming in from outside, but there are things in the built environment that we can do.
Translational perspective, I'm curious either with honestly, with any of the projects that you've described, um, could you share examples of partners that have helped you translate your work into applications? And I'm going to do a two-part question here, which is terrible because I'm not supposed to do it with surveys or any kind of research two-part questions. But the second part of that is any strategies that you have found to help orient your partners as you're trying to make that translation.
Okay. So I'll first start with the beginning of the question and give a few examples. So one example from my postdoc or from my PhD work is that we partnered with practitioners who were going into people's homes and doing mold testing. And so we had them take mold samples for us. And then I built a model using the microbial community to help determine, is the home moldy or is it not moldy? And the idea of using the community for this is that it's often really hard to tell if a home has mold damage or not. There's a lot of art to what building inspectors do when they're trying to determine if a home has particularly hidden mold damage. And continuing on that work, I'm working with partners at OSU to turn that into a test that actually would be commercially available. And so we're considering both using specific taxa, but also using specific gene expression. So as I presented, that there's all these changes in gene expression that are happening. And if some of those are signals that you have a mold problem, then you could do a test, swab a door frame in a few door frames in your house, and then get a more simple answer about whether or not there is mold damage. And so that's one project that we're working on, directly translating it into something that practitioners could use. And then if we're thinking about from the health perspective, then it's something that a doctor could ask someone to do if they're worried that their patient is living in an environment that is unsafe for mold damage, for instance. And then thinking about the HUD project that I mentioned, the idea is that if we can point to particular interventions that are improving the built environment, then we can inform policy outcomes. Because if we know that certain environments are worse, then you can have legislation to protect people to ensure that their home environment is healthier. It can also be a tool for patients and their healthcare practitioners to suggest people do in the meantime, because legislation is often slow. And especially in the built environment, it's been really challenging to have legislation because of privacy concerns. And then the second part of your question, can you say that again, Sarah?
Any challenges or strategies you've had on kind of sort of translating your work for your partners?
Yeah, I think one challenge has been just the speed of research is very different than the speed that you want to sometimes have things be implemented in practice. And so one thing that we found really important with community partners is to make sure that when you're communicating is what is the timeline, thinking about setting expectations in a way that's helpful. And that's something that I've found is one of the bigger challenges.
Awesome. So pivoting a little bit, I see a question from Roberto Alers Velasquez in the chat. He asks, when moving from single species experiments like Aspergillus to whole indoor communities, how do interactions between species influence expression and their responses to moisture?
That's such a good question. And I really wish that I had an answer. Right now, we know that in these communities, we're seeing these changes, but we haven't had a chance to dive into more simplified communities. So we've done the pure culture work, and we've also done these really complex dust communities where you don't necessarily know what's there. One of the other parallel things that would be really interesting, and I don't know of anyone doing, is doing sort of mock communities where we're putting together a smaller number of species that you know of. And then you could begin to look at the interactions between species. Are fungi talking to each other? Are the VOCs that some of them are producing, are they influencing what other either fungal or bacterial communities are doing? Is viral infection changing what's happening? We know that in water systems, some of the bacteria are more toxic when they have a viral infection, but in the built environment, no one has been looking into that. So stay tuned. I hope that maybe in a few years we'll have a better answer to that question, though.
Hang on, let me unmute myself here. Um, Super exciting comment in the chat from Seema Shargi about opportunities for collaboration and contact information. So
Oh,
thank you so
amazing.
much for sharing
Yeah.
that.
Yes.
Um, Other questions from the group?
I have a question.
Go for it.
Thanks, Sarah. Hi, Bridget. Nice to meet you. My name is Emily Hamburg Shields. I'm an OBGYN at UH. I was wondering if you could speak to like questions of acuity versus chronicity in exposure. For example, like I'm really interested in pregnancy complications and how those might result from environmental exposures. And, you know, I think it's really interesting, and often we think of as, pregnancy as a window to future health for both
Yes.
the pregnant person and their offspring. And so I often wonder, how many of pregnancy complications might be attributable to more acute exposures that are occurring during that time window when we're looking very closely and ascertaining very closely versus how many might be that we're, unmasking a susceptibility that was set up by, lifelong or years long exposure, in the preconception phase. And I'm sure this applies to many other questions of chronicity or even to like what Sarah thinks about with the origins of childhood asthma. So I was wondering if you could speak to some of the challenges with that and perhaps um, how helpful is a cross-sectional ascertainment of a microbial community in a single space and time versus, do you need to ascertain over multiple time points?
Yeah, I would say this is a big debate in the indoor environment, just in terms of how we collect samples. Because if you're thinking about looking at the microbial community, you can do like a short window air sample where you get a sense of like over an hour, what were people exposed to begin to get at those acute exposures. And then also sometimes people will do long-term settled dust over the course of a few months. But that doesn't give you any window about the variation over that time. Like maybe part of that is from like a single event that's more of the problem. And so you mentioned for maternal health and well-being. And so that's definitely it's really hard to disentangle it with many of our projects where we sometimes it's easy with measuring temperature or humidity to get real-time data, and we can see like what's happening over the course of a few months in a home or even longer if we're doing longer term monitoring. But with the microbial exposures, we kind of have to pick and choose what we're doing for sampling. And so very often we'll do just like a single sample. And so I think it is a really good question of, is that going to give us a good window of what is currently happening with asthma symptoms? For instance, if you take a set of samples over a few months or a year, or if you come back to the same home like every year, like are you going to get enough information to be able to answer those questions? I think as sequencing technologies get better, as our monitoring technologies get better, then I hope that that's going to allow us to better separate out like acute events from more chronic exposures. But I'm curious, Sarah, to put you on the spot if you have any further thoughts, particularly with asthma.
Oh, I 100% agree about the challenge about that kind of that point, relating point in time, recognizing the how dynamic exposures and experiences are. And I agree with you that it's a sampling question and a data question, right? Because those your data sets can get really huge, really, really fast, and balancing what's signal, what's noise, what's going to be meaningful, and what's going to hide something else, and cost and funding and all of those pieces. So not a particularly articulate response to that, but I think I'm really excited to see as our technologies get better and some of the innovations around how we do some of this more applied sampling improves, that we can become more sophisticated in dynamic models as opposed to cross-sectional analysis.
Yeah, I think that's another thing that's going to become really relevant with the shoe sole samples that we're doing is focusing on a particular path where we like know exactly what they're doing. Like, is that separating out what that exposure is from everything else that's happening? Or if we take a person's shoe sole and clean it to see like every few weeks what they're being exposed to, that's just going to give us a very different picture of what's happening. I'm curious, Emily, if you have anything else that you've been thinking about with your work.
Not particularly. This is why I use animal models. It's underscoring my commitment to that at this point.
Yeah, yeah.
Fair.
It's easier to control what they're doing than it is with human subjects.
Absolutely.
For that reason.
Christine, I see your hand is raised.
Hi, I'm Christine McDonald. I'm from the Cleveland Clinic. Some interesting things that you've brought up. Have you considered adding into your analyses not just microbial sequencing of what's sticking to the shoes, but what a person has antibodies to? So, for example, we do work in IBD, and individuals that are more prone to develop IBD often have antibacterial, antiflagellin antibodies that show up before they develop disease. Is there any thought about incorporating some of these measures of systemic immune response to complement the microbial response exposure data?
So I hadn't thought about that much, but that's one of the things that's so cool about these events is getting ideas like that. Right now, we've really just been thinking about the microbes because it's easier and straightforward. And we're like, do we see differences? And then the hope is that we can expand it out to begin to look at health impacts. And that'd be like looking at the specific antibodies is a really interesting idea there. And were you thinking about two specific organisms? Are there more like survey ways of looking at what antibodies are there? I have very little background in that, so.
Right, right. So I mean, I'm not an expert in this area either, but there are serology panels that are in use and some that are being more developed to look for antimicrobe immune responses, either IgG or IgA, as well as some tetramer technology to look for microbe responsive T cell populations as well. So that you have a lot of variables to begin with. I don't know how you keep them all straight, but it may be interesting to look at, which microbes are actually triggering a response in people because you can be exposed to a lot,
Yes.
but I don't know how many of them you have sort of intimate exposure with.
Yeah, and that's definitely a question when we do all of the asthma work indoors is if we're studying children who it's not the fungi that's in their home isn't relevant to their allergen trigger, then the effects can be very confounded. And there's earlier work looking at aller different types of asthma and the microbial communities, just looking at who's there. And it's very different based on the type of asthma, the effects, where some asthma, it's protective, having certain groups of fungi, for others, it's harmful. And so that gets into what you're talking about, how complex these interactions are and why it's really important to tease these things apart.
The floor is open for the group, so keep the questions coming. And Roberto Alvarez Velasquez, could per-spore toxicity or allergen expression eventually be incorporated into exposure or risk assessment frameworks? What are your thoughts on that?
Yes, that is one of our goals to be able to do that. Right now, we don't know specifically what we would want to measure to incorporate into some of the risk assessment models. We had some interest in QMRA or like quantitative microbial risk assessment and being able to, if we're able to identify some genes that are particularly important or these allergenicity panels into that, I think it'd be really useful for also just understanding like what doses are problematic. Right now in the built environment, people will find that sometimes having higher levels of fungi is more protective and like medium levels are harmful, which seems contradictory on surface value. So understanding why those different fungal exposures are so different would be really important for understanding like the true risk.
Dr. Ghosh.
I have a quick question for both of you, actually. This is really, really interesting. I'm wondering, could you talk a little bit more about what the practical application of both of your work could look like. I was particularly interested in Dr. Ronas, you talked about schools, school going kids, and Dr. Hegarty, that image of all the microbiomes in a room with the dog and the kid, like that was really also, kind of jarring and interesting as well. Given, the sort of policy implications and policy discussions about air quality in homes, especially people from, in low resource environments, I'd be really interested to hear more from you both about the practical application of both of your work.
Mhm. Yeah, I think that one high level thing is just changing how people think about microbes in the built environment, because right now we really focus just on pathogens and how they're harmful, and recognizing that some microbial exposures are good. And for that to truly be something that's well communicated, we do have to better understand what the difference is between harmful and beneficial exposures are. And that's something that my research is really trying to understand. More specifically, with my work on understanding with HUD to understand, do these low-cost interventions work? Then if we're able to find specific interventions that are more helpful, especially low-cost ones, then that's really helpful from a practical standpoint, because then we can recommend those as something that families can do, or we can start to try to have programs to help fund them, especially if we're working in low resource areas, which is oftentimes where these microbial problems, especially fungal problems, are. It's where you don't have air conditioning, where there are other like socioeconomic problems. And so having the science could help drive those changes to have better health. Like we know right now that mold is bad, but like knowing how to deal with it is part of the problem. Yeah, Sarah.
Yeah, I'm smiling from ear to ear, and not to steal Dr. Haggerty's thunder at all, because this is really her forum. I'm coming at the work from so if Dr. Haggerty is starting from the bench basic science mechanisms of action element of the translational science spectrum, which is no longer a linear spectrum, and now that lovely little like atom figure, I'm coming from almost the opposite end, from the applied clinical population level. And I got into the work because I've always, as a primary care pediatrician, I've always been really interested in care coordination for low-income kids. And what are the things we need to do to get them to optimize their health so that they can do their kid functions, which my parents always said, when you're a kid, your job is to go to school. And so if you're not going to school, why are you not going to school? So we came from the sort of end user perspective, talking with parents, with educators, with school nurses, with clinicians in the community, and then kind of worked backwards to actually sort of a similar point, in that if families are navigating challenging socioeconomic status, conditions, difficulties in terms of their built environment, the condition of their home, particularly if they're renting and don't have direct control themselves over amelioration or modification, if their child's asthma is out of control and they don't feel that their child is safe, in that their exacerbation will make it worse and their individuals aren't prepared to respond, then they are less likely to send them to school. Or if they're concerned about the built environment at the school itself. So there's been some really fun, fascinating intersections. There's the science, there's the public health, there's the policy applications. Bridget and I got connected in the first place because of one of my team members had heard her talk, like, we gotta like put some intersections together. And then there's a spinoff on my project right now, working with American Lung Association and the Cleveland Department of Public Health indoor air quality team, that as a, not necessarily as part of our research, but as a spinoff piece to get Cleveland Metropolitan School District to participate in the Clean and Clean Challenge and to do some indoor air quality work in CMSD buildings, which obviously with the kind of, and it also I think will be informed by the district changes that are coming in the coming year, in terms of being able to consolidate resources and be more attentive to the physical spaces in which children are spending their school days. And then not to talk too much, as a clinician, I also was reflecting on your conversation or your presentation, Dr. Hegarty, about have a lot of conversations with families clinically about it's not your imagination. Allergies are getting worse, pollens are getting worse, and we're in a particularly tricky part of the country geographically because of our high humidity. And we're seeing that across the board. Though I don't know that I've been as intentional about conversations with families about humidity in their own homes and indoor air spaces. Um, and I was wondering if you could talk a little bit about like whether it's urban, rural, or geographic in your work you've done and some of the either observations or take-home lessons you've had from that in terms of that humidity piece of the puzzle.
Mhm. Yeah, so right now the humidity work has been like very specific geographies. Like we've gotten dust from Ohio, we've gotten dust from Connecticut and a few other places. And the trends that we're seeing in terms of like the increase in certain of the allergen genes, also some other genes just related to fungal growth, are seen across these geographically very separate, but maybe climatically pretty similar. And so one of the things that the collaborator, Karen Dan Miller, I mentioned at OSU, is interested in is she's collecting samples from across the US to see, she's found some of these markers. Are they similar across all of these samples? Because the fungi that they're being exposed to are going to be very different. And so it's helpful in Cleveland if we have a test that we can use in Cleveland, but it's really hard to use that outside of Cleveland if it's completely different in Columbus even. And so that's something that we're trying to answer there. Another point just about like the connections between the work and trying to do that is some of the people from Sarah's project have then signed up for our project. And that's one reason that HUD is such a great partner. And it's really unfortunate that they're not continuing to fund much research because they will pay for these home remediations. Most other agencies don't want you to give money for like just doing mold remediation. And so that makes it really hard to test these things. And so that makes it hard to make policy changes. And so finding the right funding is also really important. And it also helps with the community engagement piece because if the participants are going to have the mold problem addressed that you find, then it makes it much easier to go and actually work with them because you're not just telling them that they have a problem and then like, what do they do with that information? We can actually like help them resolve it.
Thank
So
you
very,
both.
very much in line with that ethical screening kind of framework, thinking through asking questions, but being thoughtful about what we're going to do with the information. Following. You mentioned just now that like not interested in funding just mold. Could you talk a little bit about either sort of like history of the policymaking in that space or sort of the underpinnings of that comment?
Oh, yes. So HUD historically has funded both like research, which is what the funding that I have, especially about like litter removal, just healthy homes. Broadly, mold is just one piece of that. And that funding is gone now. So they're continuing to fund the projects, but they're not going to be funding new ones. So hopefully that will change with time, but there's just less interest in these areas. Yeah, and I think historically HUD has been at an interesting intersection because they are such a their mission really is about like having healthy housing and ensuring that. And so the research piece has been a small part of it, but it's been a really important part of informing what they're also doing on a policy framework.
Hi, Bridget, this is Shannon Switkowski. Thanks for presenting today. My question is, could we talk about some lessons learned, especially from the COVID-19 pandemic? Scientists are warning now with the temperature change and the next pandemic could be fungal driven.
Yes.
What types of things are we learning from that pandemic, which was viral? But we could probably learn, take some of the gems from there and apply it to this situation if that were to happen.
Yeah. So One thing that I've really noticed, and I hope will be a takeaway from the COVID pandemic, is how important the communication between public health experts is and the public is. I think with the pandemic, we saw cases where, especially in the beginning, the messages changed very quickly because we didn't want to say what we didn't know, and we were using old knowledge to inform what we were doing for a new problem. And so something that I really hope will happen, especially if it is a fungal pandemic, which would be like very different in terms of how we have to handle it, is to have better communication and to learn from some of the challenges that we had with communicating the information about the COVID pandemic. And I think that will be challenging because of policy and like political differences, but I think we have learned some things not to do from that. In terms of the difference between viral and fungal pandemics, I think that how they spread will probably be quite different. And I don't think that I can talk enough about that, but I hope that there's others who are learning a lot more from essentially the lessons of not just the COVID pandemic, but also the 19 in the early 1900s, other pandemics that we had with like the bird flu. And especially to take the lessons where across the country and across the world, we were doing very different things and handling it, like which ones worked, and using that to inform our policies and not just continuing to do the same things.
Are we seeing a lot more research on new antifungal medicines or treatments?
There's definitely a lot more interest and funding onto just like understanding these fungal threats. I don't know, like right now we don't have enough good antifungals and we don't have good treatments for them. So there's a real interest to develop those to hopefully get ahead of some of these really scary problems because and it's not only like the fungal threats that are really problematic. Some of my other research is looking using phages as an alternative to antibiotics, and there's increasing antibiotic resistance. That's a big problem. And that's separate from climate change, but it is a big environmental health issue because there's the interchange between the environment and human that is leading to lots more antibiotic resistance.
So, given all of these emerging questions, as we come to the top of the hour, do you have advice or recommendations for folks who might not have any kind of background or grounding in these fields but want to get involved in this type of research?
Yeah, I think that's a great question. I think it depends a lot on what your question is. I think if you have microbiomes questions, then it's reaching out to individuals like myself who maybe don't have the medical background to form partnerships because there's lots of excellent people in biology and environmental engineering who are thinking really deeply about microbiomes separate from like the gut and human health that could lead to very informative partnerships. And I think it depends, like, is your question about maternal health? Is it? Then the specific questions will be very different than if you're more interested in other chronic diseases.
Well, I just want to say thank you to both Dr. Ronas and Dr. Haggerty for their time and their experience today. Of course, my screen loves to share and work in this frozen weather, but we invite you to join us next month at our Science Cafe session. You can register. There will be an email sent out with the recording. And we just appreciate your time and we hope you all stay warm today. So we'll hope to see you at a future event. Thank you so much.